This is the second lecture in the series on the cardiovascular system, specifically blood composition and function. In this lecture, we need to go through the composition of blood, so the cellular components and the non-cellular components. We need to explain about how transport of gases by blood components occurs, and we need to discuss some various disease states which we associate with the blood. When you take your whole body weight, approximately 8% of that weight is due to your entire blood component. The remaining 92% is due to other fluids and tissues of your body. If we look at the whole blood just for the moment, it's split almost equally, 55% blood plasma, 45% formed elements. Just looking at the blood plasma for the moment, plasma consists of 91.5% water, and the remaining 8.5%, 7% of that is protein and other solutes such as electrolytes, nutrients, gases, regulatory substances and waste products accounts for the final 1.5%. Of the formed elements in blood, 45% total volume consists of platelets, white blood cells and the red blood cells. The red blood cells are by far the most numerous of these formed elements. The white blood cells can be further subdivided into neutrophils, lymphocytes, monocytes, eosinophils and basophils, the most numerous being the neutrophils, consisting approximately 60 to 70% of the white blood cell fraction, with the basophils being only 0.5 to 1% of the entire white blood cell fraction. If you ask somebody to look at hematocrit, they're looking at the percentage volume of your whole blood which is occupied by your red blood cells. This is performed by taking a blood sample and centrifuging it down to compress all those blood cells down at the bottom. You usually see a line of red cells with a fine white line on the top. That is the white cells, the platelets, and then the blood plasma is separated out at the top. By identifying the percentage of the volume of whole blood occupied by the red cells, clinicians can tell if a person has forms of anemia. If we look closely at the blood plasma, the vast majority of the proteins contained in the plasma belong to the class of albumins or globulins. The proteins in blood plasma are large non-penetrating solutes and maintain the osmotic pressure. They're also involved in buffering the blood pH, binding transport, so things like lipids, hormones, vitamins and drugs can bind to the proteins within the plasma to be transported. Proteins within the blood plasma can act as enzyme precursors and cofactors, or can act as signaling molecules. A good example here are hormones. In addition, we have the antibodies. These are immunoglobulins released from the B lymphocytes. This is one of the class of white blood cells. They're involved in the immune system. Another important class of proteins we find in the blood plasma is fibrinogen, which is critical for the blood clotting cascade, which we will come on to later on. In relation to inorganic ions in the blood plasma, the key list of ions are shown on this slide. They control the amount of water in the blood and extracellular fluids by osmosis and exchange of ions, for example, sodium potassium ATPase. They can also act as buffers, enzyme cofactors, involved in membrane excitability, such as again sodium potassium chloride, blood coagulation and clotting. Potassium is a key ion involved in the blood clotting cascade. The gases dissolved in blood plasma are oxygen and carbon dioxide, and we also find nutrients and nitrogenous wastes also dissolved in the plasma. For example, glucose, lipids and amino acids for the nutrients, urea, ammonia and uric acid for the nitrogenous wastes. In addition to these, we can also find some regulatory and signalling molecules such as hormones dissolved in the plasma. These are in addition to those which are bound by the protein fraction. Moving on from the plasma fraction and looking at the cell fraction of whole blood, the process at which cells develop into the blood cells is called hematopoiesis. In this case, if you look at the top, you'll see a pluripotent stem cell. This is a stem cell capable of developing down several different lineages, but it is not capable of developing to any type of lineage. In this case, it can either develop down a myeloid stem cell pathway or a lymphoid stem cell pathway. If you follow the lymphoid first on the left of the slides, you will see that there are some green boxes. These are the colony forming units. These are the hematopoietic growth factors, and these are what drive that stem cell to develop down particular cell lineages. For example, the colony-forming units for erythrocytes push the myeloid stem cell to develop to proerythroblasts. At some stage in this development, the proerythroblast becomes a reticulocyte. As the reticulocyte matures, it ejects its nucleus and becomes a mature red blood cell or erythrocyte. Next to that, the myeloid stem cell, if exposed to colony-forming units megakaryocyte, will become a megakaryoblast. The megakaryoblast will develop further into a megakaryocyte, which then buds off to form platelets or thrombocytes. Alternatively, in the myeloid stem cell lineage, cells can either become eosinophilic myoblasts, which will develop to eosinophils, basophilic myoblasts, which develop to basophils, or, if exposed to colony-forming units granulocyte macrophage, can either develop to a myeloblast or a monoblast. The myeloblast will become a neutrophil, and together with the basophils and eosinophils, these three cell types form the granular leukocytes. If, however, a monoblast is formed, that will develop into a monocyte. Monocytes are unusual cells because they have the ability to actually migrate out of the blood circulation and develop into mature macrophages. These are phagocytic cells of the immune system, and we will come on to them later. Alternatively, if the pluripotent stem cell has developed down the lymphoid stem cell lineage, the cells will become either T lymphoblasts, B lymphoblasts, or NK lymphoblasts. Those cells will mature to T lymphocytes, B lymphocytes, and natural killer cells respectively. Monocytes, T lymphocytes, B lymphocytes, and natural killer cells form the agranular leukocyte fraction. These are cells that do not have granules in the cytoplasm. B lymphocytes, under certain conditions of the immune response, can mature and develop into long-lived plasma cells and memory cells. We can come on to this later when we do the immune system. Another good hemopoietic growth factor you need to be aware of is erythropoietin or EPO. This is used for athletes in some cases to artificially increase the number of red blood cells that they produce. Naturally, people who live at higher altitude tend to have a higher amount of EPO in their bodies naturally. They tend to have higher red blood cell counts naturally because the oxygen at high altitude tends to be thinner. As a result of having a lower concentration of oxygen in the air that they breathe, these athletes have developed a higher number of red blood cells to compensate to ensure their cells get the correct amount of oxygen. If they then come down to a lower altitude, so sea level, they effectively have a natural competitive advantage. Certain drug allegations are made involving EPO, and it is now one of the banned substances in international athletics as a result of giving athletes an unfair advantage. Now I'm going to work through each of the different cell types, starting with the erythrocytes. These are the red blood cells. They are approximately 7 to 8 micrometers in diameter and are biconcave discs with a very large surface area. They lack the nucleus. If you remember from the previous slide, the reticulocytes actually eject the nucleus during their development, and that's what gives you this biconcave disc shape. It's a simple cytoplasm. Its major component is hemoglobin, and its key function is the transport of oxygen and carbon dioxide. Red blood cells are very short-lived because they do not have a nucleus or other cellular components to allow them to survive for longer. In this slide, we can see some various different cell types. The vast majority are the red blood cells. We'll also identify a white blood cell at the top right of the slide. This is a neutrophil. And at the bottom left of the slide, you can see a white blood cell, which is a monocyte. There are platelets dotted around on this slide, and you can just about see them as very small cellular components. Thrombocytes or platelets are released from megakaryocytes. Again, like the red cells, have no nucleus, but in this case their cytoplasmic granules are present. Thrombocytes' primary function is in blood clotting, and we'll come on to the clotting cascade later. Of the white blood cells, we mentioned granular and agranular leukocytes. At the top we have eosinophils, on the left, in the middle is a basophil, and the far right is a neutrophil. You can see why they're called the granular leukocytes. Their cytoplasm is absolutely full of granules. Eosinophils are called eosinophils because they take up eosin dye very, very readily, which gives them that sort of dark pink coloration. The basophils have a huge number of granules. They are one of the smallest of the blood cells and the most dense for the granular leukocytes. They're quite easy to identify, and you don't tend to have very many, especially if you look in a blood slide. Neutrophils, as I've mentioned before, are the most numerous of the white blood cells. They have a multi-lobed nucleus and granules within the cytoplasm. The fact that they have this multi-lobed nucleus is one of their identifying features. For the agranular leukocytes, at the bottom we have lymphocytes on the left and monocytes on the right. The monocyte is described as having a horseshoe-shaped nucleus. It's a very large cell. Note the difference in appearance between a horseshoe-shaped nucleus for the monocyte and the multi-lobed nucleus of the neutrophil. For the lymphocyte, in this case bottom left shown in D, you can't tell the difference cytologically between a B lymphocyte and a T lymphocyte. They're basically small, round cells with no granules in the cytoplasm, and the nucleus is approximately 90% of the cell's volume. The main component of the red blood cell is haemoglobin. In healthy adults, haemoglobin consists of 2 alpha and 2 beta subunits. Each of the heme groups per subunit each binds 1 molecule of oxygen, and this is a reversible binding. The heme groups consist of ions contained within the heme itself. So each haemoglobin molecule is capable of carrying 4 ions. Oxyhemoglobin is the name given to haemoglobin that is bound to oxygen. This takes oxygen to tissues, where the oxygen dissociates to give you deoxyhemoglobin, releasing the oxygen to where the tissues need it. It's essential to understand with blood cells how dissociation of oxygen differs depending on the physiological parameters. This is a classic oxygen dissociation curve, showing partial pressure of oxygen in millimetres of mercury against percentage saturation of haemoglobin. The curve goes up straight through, and we have 3 points identified on the curve. Starting at the right hand side first, so the situation of oxygenated blood in the systemic arteries, at 100 partial pressure of oxygen, so 100 millimetres of mercury, we have just under 100% saturation of haemoglobin. That's the situation you'd expect. This is blood that is freshly oxygenated, fully oxygenated, so it's going to have maximum saturation. Towards the middle of the curve, we have deoxygenated blood in the systemic veins. So this is blood that has passed through the capillary beds of the tissues and is now in the systemic veins returning to the heart. If you look at any given partial pressure of oxygen, so in this case, say 40, the percentage saturation of haemoglobin has dropped to 75%. That shows that there's still a considerable amount of oxygen available in deoxygenated blood. However, it has decreased from the value we saw in the oxygenated blood in the systemic arteries. Finally, furthest on the left of the curve, deoxygenated blood located in contracting skeletal muscle, so for example with exercise, partial pressure of oxygen is only 20 millimetres of mercury. The oxygen has been used up very rapidly by the tissues, by the muscle in this case. When you take that across to see the percentage saturation, you find it comes out at 35% saturation of haemoglobin. So for the lower the partial pressure of oxygen, the lower the percentage saturation. That means that as your body requires an increased delivery of oxygen, and by doing so drops the partial pressure of oxygen, the amount of oxygen that is released from the haemoglobin will increase. Oxygen can reassociate with haemoglobin once it is disassociated from it. So there's a particular substance called 2,3-diphosphoglycerate which stabilises deoxyhemoglobin, increasing the amount of oxygen that is actually delivered to the tissues. The oxygen dissociation curve can shift depending on physiological parameters. So for example, if you're doing a lot of exercise, you're creating a lot of CO2. That would mean that your blood pH will become more acidic, it will drop. Your body temperature will increase, and you will increase the partial pressure of carbon dioxide. In doing so, the line moves to the right. That means that for any given partial pressure of oxygen, you will release more oxygen from the haemoglobin, so percentage saturation of haemoglobin will decrease. If, however, you are not doing any exercise, and for whatever reason your blood is becoming more alkaline, so increased pH, decreasing body temperature, and decreasing PCO2 levels will move the graph to the left. So for any given partial pressure of oxygen, there is a higher percentage saturation of haemoglobin, so the blood is less inclined to release the oxygen. In other words, in this situation, systemic capillaries, the haemoglobin would rather associate with CO2 than oxygen. Chloride shift occurs, so this time we're taking chloride ions from plasma into the red blood cell. In addition, the CO2 that's taken into that red cell will interact with water, and the actions of carbonic anhydrase will become H2CO3, which will dissociate into bicarbonate ions and protons. The protons will then remove an oxygen from the haemoglobin, releasing an oxygen into the plasma. This then diffuses across the systemic capillary wall into the interstitial fluid to be used by the tissue cells. The bicarbonate ion that's produced from this reaction diffuses into the plasma and again is there to maintain the buffering of the pH. Various disorders of the erythrocytes are recognised. The first and probably most common is anemia. In this case, it's a decreased ability of blood to carry oxygen. There can be a dietary deficiency of iron, a deficiency of vitamin B12. If this happens, it's called pernicious anemia, which is a slightly different condition. Deficiency of folic acid can also reduce the number of red blood cells we produce. And bone marrow failure due to cancer or cytotoxic drugs can also result in quite severe anemias. One of the more worrying of causes of anemia is hemorrhage, so blood loss can be internal bleeding or external bleeding due to severe injury, which naturally will reduce your blood volume and the amount of oxygen you can carry. Disease of the kidneys will also cause anemia, as you end up with a lack of secreted erythropoietin. If you remember from the original slide showing hematopoiesis, erythropoietin is one of the essential growth factors for development of red blood cells. Alternatively, the red blood cells may form perfectly fine but be destroyed in the body. This is hemolytic anemia. Alternatively, you could have a genetic disorder. In this case, the most common of the genetic disorders that result in anemia is something we call sickle cell trait. This sickling of the red blood cells tends to occur under states of stress, physiological stress. There seems to be a high prevalence in African, African American, Mediterranean, Middle Eastern, East Indian, Caribbean and South and Central American populations, so basically pretty much every population other than Caucasian and North European tend to see a high prevalence of this condition. The problem with sickle cell anemia is that it significantly affects how the blood can carry oxygen, but it also causes severe pain. Once formed into a sickle shape, these cells can't move as easily through your blood vessels. They tend to block blood vessels, leading to a situation called sickle cell crisis. This causes extremely severe pain. Damage to tissues usually occurs at junctions of blood vessels, so where your blood vessels are splitting into narrower arterioles than capillaries, so you're going to have major problems in various organs, including the brain, heart, any of the major organs. The red cells in sickle anemia have a shorter lifespan than normal red blood cells as well. So not only do they fail to bind as much oxygen, but they don't last as long either, so that leads to a double anemia. It isn't actually entirely bad, however. If you have sickle cell anemia, you basically are homozygous for the HBS gene, which we believe is a codominant gene. If you are sickle cell trait, it means you only have one copy of the hemoglobin S gene. That means that only some of your cells will form sickles, the rest are normal. You will still have a certain level of anemia, but not as severe. You are less likely to undergo a sickle cell crisis. But the key importance here is that you have a resistance to malaria. If you think back to the major places where we see sickle cell disease and high prevalence, a lot are in areas that have severe problems with malaria. This is the body's natural defence. For some reason, the malarial parasite cannot survive in sickle cells. We don't know why. So that's a disorder of the red blood cell. A disorder of the white blood cells are slightly different. You have leukopenia, which is a deficiency of the leukocytes due to suppression of bone marrow function. Usually we see this occurring as a side effect of cytotoxic drugs and radiation damage. Alternatively, there's leukemia, which you will all have heard of, which is an uncontrolled production of blood cells due to bone marrow tumours. You can have chronic and acute forms of leukemia, and depending on which lineage those cells have come from determines the name we give it. So a lymphocytic leukemia, generally we see an excess B cell precursors. In myelogenic leukemia, we tend to see cells of the myeloid lineage, so erythrocytes and granulocytes. As a rule, in leukemia, it's not just an uncontrolled production of these blood cells, but they are not forming correctly. So we see the blast stage cells coming out into the circulation. These cells have not finished developing yet, they are not functional. As a result, we see significant immune suppression in these conditions. In summary, your blood then is composed of plasma and different form elements, each of which has a specific role. The erythrocytes bind and transport oxygen and release it when demand is high, and conversely will bind and transport carbon dioxide, releasing it to the lungs to be removed from the body. Anemia is a state where we have a lack of functioning erythrocytes and can be due to various conditions or predisposing factors. And leukocytes form part of the immune system which defends the organism against infection, but we will cover far more of that when we do the immune system lectures. As always, if you need any help, please do not hesitate to contact me. Probably best to contact me on email on the information shown on this slide.